So, let us start today's class. As we discussed in the last class, today we will talk about insulin chemistry and its physiology. As I mentioned in the last class, insulin is a peptide, and if you look at the empirical formula of insulin, it has 254 atoms of carbon, 377 atoms of hydrogen, 65 atoms of nitrogen, 75 atoms of oxygen and 6 atoms of sulfur, and it has a molecular weight of 5734, right. Insulin is built from 51 amino acid residues and is one of the smallest protein in the body. It is structured, and while we are talking about the smallest protein in the body, mind it was Collip who helped us to understand how to isolate insulin from a mixture of pancreatic secretion. It is structured with 2 polypeptide chains linked by 2 disulfide bonds. These disulfide bonds are these sulfurs which take part in it. Connecting the amino acid cysteine to cysteine, there is also a third disulfide bond that connects those same amino acids within a chain A. Chain A consists of 21 amino acids and chain B consists of 30 amino acids. So, that makes it 30 plus 21 as 51 amino acids. The one special thing about insulin is its change structure to become useful in the human body. Insulin is originally produced as pre-proinsulin, which is transformed to pro-hormone molecule by proteolytic action into proinsulin and finally into active polypeptide hormone insulin. So, our take home about this understanding is that insulin is not produced as it is. Insulin has lot of protective sites before it comes into action. So, that means whenever we are talking about an extract, you have to ensure that insulin is in active form. Why I am telling you this? When there were some experiments where the first clinical test on human patient were conducted on severely diabetic 14-year-old boy. Although the injection of extract failed to have resoundingly beneficial effect, Toronto team continued the experiment. A short while later, Collip made a breakthrough in purifying extract using alcohol, slightly over 90% concentration to precipitate out the active ingredient of insulin. This word, active ingredient of insulin, or active ingredient which is insulin. So, what if this experiment, the insulin was not in its active form, as in the pro form or prior to pre-proinsulin likewise. So, you always have to realize that before you write off an experiment, you have to think multiple times that why it is not working outside the body, because you are extracting this fluid from inside animal and then you are injecting it into another animal, which is a very arduous task to get exactly the same response. But then these are the challenges you will always face whenever you are doing experiment of this kind. Okay. So, if you look at it, insulin is originally produced to become useful in the human body. The one special thing about insulin is its change in structure to become useful in human body. Insulin is originally produced as pre-proinsulin, which is transformed into prohormone molecule by proteolytic action, means breaking it into proinsulin, and this proinsulin eventually become insulin. So, this is a multi-step process. If you look at the structure of insulin, this is how the structure looks like. You have the disulfide linkages sitting here, you have another disulfide linkage sitting here, accounting for the 2 disulfide linkage, and then you have another disulfide linkage out here. So, essentially these disulfide linkages account for your sulfur 6. If you, as I mentioned, there are 6 sulfur, and if it is a peptide, then the only way you could have 6 sulfur could be 6 amino acids containing sulfur, and sulfur containing amino acids are cysteine and methionine. In this case, it is all cysteine. So, here is one cysteine, 1, 2, 3, 4, 5, 6. These are the 6. So, as I mentioned in the previous slide, there are 2 chains, chain A and chain B. Chain A is connected to chain B by 2 disulfide linkages, and chain A, within chain A there are disulfide linkages which helps it to curl on its own, something like this. And this structure is of immense significance, as this is the active structure. Now, if we look at the proinsulin structure, another look at the image of the proinsulin made of 86 amino acids, while this one is 51, proinsulin is 86. So, it means there has to be some chopping of some of the moieties, which and again you see here is the disulfide linkage and within, and then this is the part which gets chopped off. So, it means there is a specific cleavage sites which will cleave this part of proinsulin. And if you look at it, the process of pre-proinsulin to proinsulin to insulin, this is how it is converting pre-proinsulin. Pre-proinsulin is 110 amino acids, and in the previous class we told you that this is 86 is proinsulin, 86 amino acids. From here we are moving on to 51 amino acids. Think of it, how lucky those 4 people have to be that they could exactly get this, the active form. Rest all are not active forms. Realize that this is extremely important for you to understand. Converting proinsulin to insulin, pre-proinsulin is transcribed as 110 amino acid chain, and by the removal of the signal peptide, proinsulin is produced. Formation of disulfide bond between A and B chain component is made. The removal of the intervening C peptide chain produces biologically active insulin of 51 amino acid. Okay? So, this is how it looks like. So, this is your pre-proinsulin, this is your proinsulin. Now, what you have are, this part is removed, this C chain is removed, disulfide bonds are not here. So, at pre-proinsulin there is no disulfide linkages, followed by establishment of disulfide linkages, and here the removal of the C chain. And all this process of understanding happened much later. It was not in 1921. 1921 or 1923 was the beginning of understanding, or I say the beginning of the modern era of insulin, prior to the discovery of insulin and its use in treating diabetes in human. That brings us to a point how really the blood glucose level is being maintained by insulin. So, the physiology is like this. You take food containing sugar, or we call as carbohydrates. Okay? It travel along your gut. So, this is the large intestine. Once again, this is the large intestine, This is the small intestine, stomach, and so on and so forth. Okay. Now, what happens is that when you take sugar or carbohydrate, the sugar and carbohydrate is the readily form of available energy. Readily available for energy. Okay. Now, these carbohydrates are being absorbed from the food by the blood. This absorption process is being facilitated by insulin. So, insulin is kind of a gatekeeper. It helps to pull the glucose from the blood vessels, and it is used for storage and convert it to higher form of carbohydrates or complex form of carbohydrate, or use it immediately for energy. So, if insulin is not present, what will happen is your blood will have higher concentration of glucose, and if your blood has a higher concentration of glucose, automatically by the rheological properties, the density of the blood will increase. So, it will put more stress on your heart to pump the blood, because blood becomes heavier. And what it will do is that it will put lot of stress on your kidneys to throw away this glucose molecule. And if you look at some of the initial work of MacLeod, he worked on glycosuria urine. So, basically he was saying the diabetic patient has lot of glucose or carbohydrate in their urine. So, during diabetes, your blood pressure goes up because the body has to pump more, because your blood gets thicker or the fluidic property becomes more denser because of the presence of the blood glucose, because insulin is not there. So, body cannot absorb the insulin from the blood. Further, it affects the kidneys because kidney has to throw away the glucose in the urine, and the kidney has to function more. So, what is happening essentially, because of the absence of insulin, in the case of diabetes, your heart functioning is compromised, your kidney is compromised, and your energy level is being compromised. Right? So, when there is a high level of glucose in the body cells, the insulin is secreted, then glucose is taken over. Insulin allows the glucose to be absorbed by the body cells, and the blood glucose level reduces. The pancreas produces insulin which enters the. So, it means there is a clock. The clock says, okay, there is a higher concentration of blood glucose level in the blood. So, pancreas, you secrete insulin, insulin will go and go to the specific organs and will bind and will open the gate for the glucose receptors. So, the glucose will be taken over by those different tissues in our body from the blood, right. So, in a way, insulin is acting as a gatekeeper. Insulin goes and binds. So, once again try to understand, you eat a food, there is an increase in blood glucose level, pancreas senses it, pancreas secretes insulin. Insulin goes and bind to different organs where this glucose is being absorbed. So, in a way, insulin goes and bind to those different organs and open their glucose gate. And from the bloodstream, these blood glucose is being absorbed by these different organs. So, insulin is a gatekeeper. Try to understand. And then, of course, we'll go to diabetes type 1, type 2. You'll realize where it exactly works. Further, in the case of low level of glucose, say for example, your body has low level of glucose, what will happen? Insulin will be not produced by the pancreas, less glucose absorbed by the body cells, blood glucose remains the same, and normal level of blood glucose remain. And there are other hormones which are involved in that process too, of the breakdown of the stored glucose molecule or stored carbohydrate molecules to release, to help in withstanding low glucose level. So, what we see here is that, say for example, if think of a situation, your body has a low glucose level, say for example. In this low glucose level, if I inject insulin, what will happen? And this is something we will be discussing. This is a catch, and I will discuss about it very soon. Okay. So, if we look at the control of blood glucose concentration by pancreas and insulin, what we see, glucose is needed by the cells for respiration. The concentration of glucose in the blood must be maintained at a constant level and controlled carefully. Insulin is a hormone produced by pancreas which regulates blood glucose concentration in the blood, as I mentioned just a couple of minutes back. If the blood glucose concentration is too high, the pancreas produces the hormone insulin, which causes glucose to move from the blood into the cell. In liver and muscle cell, excess glucose is converted into glycogen for storage and will be used later date. So, body knows how to translate glucose to glycogen at specific tissues, but insulin is needed for those tissues to open the gate to allow the glucose to enter into them. Now, when we talk about diabetes, then we are talking about a situation where there is no insulin production. If there is no insulin production, then the glucose in the blood is extremely high. So, you need to inject. So, cells do not absorb glucose. So, that means the number of glucose particles in the blood has increased significantly, thereby influencing the flow. And of course, as I mentioned, it will influence the heart, it will influence the kidney, it will influence your urine and multiple other things in the body. You will have a lot of sweating, perspiration, body will try to balance it out in case of no insulin. So, only way you can handle this is an external source of insulin, which could take care of, which goes and binds to the adipose tissue, liver, to ensure that the glucose is being absorbed. This is one way. Now, there is another form of diabetes, which is type 2 diabetes. Cells do not absorb glucose. This is far more complex. You have no dearth of insulin, your body is secreting insulin, but cells do not respond to insulin. That means, as I mentioned, the insulin is the gatekeeper. Insulin goes and binds to the skeletal muscle, to the liver, and open the gate for glucose. But say for example, a situation where these cells do not respond to insulin, cells do not absorb glucose. This is a far more complex situation. So, these cells are no more even responding to, so there is some form of mutation or some problem on their insulin binding site which does not allow the glucose to be taken over. So, these are two critical cases of diabetes. So, this one is curable by the work of Banting, MacLeod and their students, but type 2 diabetes cannot be cured like this. It's altogether a different ball game, type 2 diabetes. Now, if you look at the current status of average out-of-pocket monthly diabetes cost, this is coming from US dollars in the March 2016, it's just 7 or 8 years back, right? In US, When I say out of pocket means even after insurance has covered you, 360 US dollars. In India, it is 112 US dollars. $100 means we are talking about anything in the range of approximately it's around 10,000 rupees per month. And this is after your insurance company has covered. That's quite a huge medical expenses. And this price goes down in Japan, it's 80 USD, UK it is 65, Italy it is 19. Cost include insulin as well as other diabetic supplies. The International Insulin and Diabetic Supply Survey by BBC indicates that. So think of the amount of expenses, and even in US 360 US dollar per month is significantly high. So these are some of the major challenges what humanity is going to face as more and more cases of diabetes are increasing. And mind it, India is diabetic capital of the world. So it is worth looking at the story of insulin and the research on insulin with more and more sensors which are coming. But our next class will be devoted on the murders which have happened using insulin, which is another gory side of insulin and how it has stirred up controversies over the years. Thank you.